one thing we could certain, generally speaking, we know the conservative sequence, what it looks like. So this is what we're going to talk about right here. What is the conservative sequence? The place started replication we call +1. Anything happened on the right side of the blackboard, we call downstream. Anything happened on the left side of the blackboard, we call it upstream. Start from the +1, about 10 base pair. Okay, that means BP. You call base pair. The base means ATGC. Here is AUGC. Okay, or ATGC. There's a very conservative sequence, you can see it, is TA, TA, AT. Corresponding to the second band is AT, ATA. That is called -10. We also give them a name called pyramidal box. Then we also find around 16 or 18 upstream, which means around 35 upstream, start from the initial point, there is another sequencing. TAGACA. TTGACA, sorry. So corresponding AA, CT, GT, this is called -35. And the distance, the whole section between -10 and -30, this is called a pyramid. This is where all your polymers start to recognize. This is an important slide. You should mark it 3-star. This tells you what is a promoter, what is the conservative sequence of the promoter, where it started translation or transcription. The conservative sequence here, you see TA18, we call it -10. That's called the pyramidal box. Around the 16-18 BP upstream is -35, is TTGACA, and this whole area is pyramidal region. So in the exam 3, if I ask you to label the pyramidal, can you label? Very simple. Look at its conservative sequence. I just mentioned that this transcription, the conservative sequence, is a universal one, which regulates the general life for the bacteria. But how about the others? It's different. This promoter we just mentioned is for general life of bacteria. That's why we call it sigma 70. What does 70 means? That's a mass weight of a protein. It's 70 delta, 70,000 DA. So sigma 70, if we want to talk about this is 70,000 delta used to measure in the molecule of enzyme. If the bacteria in the heat processing exposed to the heat, they will have a heat shock response. Their sigma factor is different. Is sigma 32. This is their sequence. If the bacteria related to the motility, which means they're really moving, and the flagella components, that's sigma 28. So just be careful. what we mentioned about the promoter here is a universal one, sigma 70. During the bacterial lifetime, they have a different sigma to combat the environmental stress and different function. Okay? This is how that goes. So we go this really easy. They're combining RNA polymerase unwinds DNA, transcription bubble comes, and they'll need RNA polymerase, and the transcription is always from 5' to 3', and they generate a bubble which is RNA DNA hybrid. Okay? And finally, how do you know the termination? There are two different characteristics will happen. Number one, when they reach an area, something this happens. This is called a stem loop. When you see these, which means the body tells you transcription stopped. Don't do it anymore, it's enough. The second thing is that you see lots of U there. That's called rich U. When you see the rich U area, stop transcription. It's more than enough. Bacterial transcription is also regulated by metabolism. We're going to talk about that later on. Not very deep. So why we say, I mentioned all the time, when you see uracil, it's not a very stable chemical. What's the uracil looks like? The difference between uracil and thymine, it's in the position 6. Thymine, you have methyl CH3, uracil is proton, which means you replace thymine at, instead of you have a methyl, you have a proton, you have a H at an unimportant place. Once this happens, become 2H. That's not a stable structure. It's very easy to happen biochemical reaction. So that's why it stopped. And you shouldn't be feeling about these. When we later on we talk about it. Hey, let's say these things. This is not stable, double bond. I mean, you need to have the feeling about it. Although we don't talk about like detailed chemistry, but this is not stable. Same thing here. Okay, transcription termination. Occurs the core RNA polymerase dissociates from temporary DNA. DNA sequence mark the end of the gene in the trailer and the terminator. Some terminates require the aids of the rho factor. They could also have a rho factor for termination. So 3 markers here. Rich U area, stem loop, and the rho factor. Okay? Could also use using rho factor. When you see these, transcription is shut down. If you don't know, this is a picture, cartoon movie picture. What will happen? Promoter is there, transcription starts. RNA polymerase recognize the promoter. So -10, -35 conservative sequence. Then the synthesis. RNA synthesis by complementary base pairing of 3 nucleotides with the nuclear base on the template DNA strand. The site of synthesis move along the DNA that has been transcribed rewinds. Transcript reach a terminal and then will be released. DNA and RNA polymerase are released and the DNA helix reforms. Okay, that's a very good picture. So that's all I have for this slide. This slide is we talk about the DNA replication and RNA transcription. What's the key here? You should always remember the story is for the enzyme function during the DNA replication. And this is a key. tells you anything at the beginning. And I really like the slides is right here. This is a very good 3 slides, 3 pictures, explain to you DNA replication. So DNA replication relatively simple. This is a 3-star, 5-star slides for transcription. We pay much attention to the initiation, the promoter, -10, -35 sequence, combined with RNA polymerase for transcription. So these couple of slides, I would say 4 slides, is the most one. You should mark it 3-star or 5-star. In the exam, we're going to use it. Okay, so that's all I have today.